Alrighty, our first lecture will be on microbiology and cell culturing. So, what is microbiology? It's the study of microorganisms and their effects. So, a microorganism is defined as something that can't be seen without a microscope. So, we always think of bacteria, but that's not the only thing. We're going to include yeast, algae, fungi, and viruses. And yes, it's true, in ninth grade, we did discover together that viruses do not exhibit all of the characteristics of living things and therefore are considered non living things. However, we treat them like they're a living thing because, in many ways, they act like a living thing. And so we're going to include them on this list. I think it's really important to understand that when you learned about taxonomy in middle school, you learned kingdom, phylum, class, order, family, genus, species. But since then, we've added domains. And when you look at the three domains that all of life fit into, two out of three, two out of three are devoted to just prokaryotic organisms. Archaea are referring to more ancient bacteria that tend to be extremophiles, meaning that they live in very extreme temperature environments or salt environments or things like that. And then the eubacteria or just bacteria, those are prokaryotes that tend to live with other eukaryotes. So I think it's really important here that we highlight the fact that most living things, most species, when you look at those three domains, are prokaryotic. So we are dominated in this world by microorganisms, even though we don't see it. We can't avoid the fact that our understanding of microorganisms came from our interest in understanding diseases. We saw that people were getting sick and we wanted to find the problem for those. And also, our understanding of microorganisms came with technology, right? In order to know these microorganisms were there, we had to be able to see them in some way. So, inventions of microscopes has been really an important part. Some examples of microorganisms that you may have heard of. Malaria is caused by Plasmodium. A lot of people think malaria is caused by mosquitoes. And while that's true, mosquitoes are vectors, meaning they carry the disease. It is the Plasmodium, which is a protist, that is within the saliva of the mosquito. And specifically, it's the female Anopheles mosquito. Anthrax caused a big scare back in the beginning of 2000. That's the powder that was being mailed to different offices around the United States. Anthrax is a disease that is caused by Bacillus anthracis. It's inhaled and it causes major upper respiratory inflammation, and that can cause some very serious issues. The Black Plague, caused by Y. pestis, or Yersinia pestis is its full name, but we abbreviate it sometimes. And tuberculosis, also another respiratory disease caused by Mycobacterium tuberculosis. Okay, so different microorganisms do cause diseases, and we have definitely started our understanding of them because of this need to understand what causes disease. So let's take a look at what these microorganisms are. They're not all prokaryotic. There are microorganisms that are eukaryotic as well. But we're going to do a quick cell review, and then we're going to talk a little bit about how they're important in biotechnology. So bacteria, single-celled organisms, okay? So an entire organism that is made up of one cell. They can be harmful, as we just saw. For example, food poisoning, right? Taco Bell has had a number of problems with these because of E. coli that can cause food poisoning. However, bacteria are very helpful to us. In fact, we've watched a couple TED Talks about how beneficial they are to us. We're going to do some more exploration about how beneficial they are. And just, here, think about the fact that they aid you in your digestion and in your vitamin production, specifically the bacteria that live on your intestines. And I say on your intestines versus in your intestines because this is something to think about. Your body is actually open to the environment on the inside and on the outside. So picture this, picture a paper towel roll, okay? What parts of the paper towel roll are exposed to the outside? The very outer layer of white paper towel, right? Once you unwrap that plastic. But then also, when you look at the tube in the middle, okay? That tube in the middle is also exposed to the outside. That very much represents your body. So when you think about it, your gut tube is open from your mouth to your anus. And so because of that, all of that area is actually outside of your body. It's a really weird way to think about it, but that's how bacteria are living on the surface of you, in addition to on the surface of your skin, your face, things like that. So remember that your gut tube, even though to you it's inside your body, it's actually outside of your body. Okay? So bacteria are not within you, they are on you. They're very small, okay? 1 to 10 micrometers. And remember, micro is 1 times 10 to the negative 6th meter. So think about how small a centimeter is, and then divide that up even further. So not milli, keep going, okay? We can't even divide it up. We need a microscope to see them. Morphology-wise and shape, they actually fall into 3 basic categories, okay? So the spherical is called the coccyx, and that is the pink round ones. Bacillus would be like your rod-shaped, and those are your yellow ones in this picture. And then sporillum are spiral-shaped, and I kind of think they look like a pasta noodle there, those orange ones. So most bacteria can be classified into these 3 shapes. What makes bacteria unique? Bacteria have a cell membrane, as all cells do, but in addition, they also have a cell wall. We do not have cell walls on our cells, but plants do, okay? And so they're similar to plants in that they both have a membrane and a cell wall. But for prokaryotes, this cell wall is very specific. The way that it is built, and I have a picture here that shows you, okay? So right here, you see your typical cell membrane made up of your phospholipids and then your transmembrane proteins. But then attached to the outside, all of this red and purple out here, this is the cell wall area, okay? And it is created from something called peptidoglycan, and it's a layer of chemicals, right? It's a layer of molecules that are all linked together. And we've noticed that there are 2 different ways that bacteria can build those cell walls. And the cell wall can be built so that it is very thick or it is very thin. And when you apply a stain to certain bacteria, you can tell the difference in them if they are gram positive or gram negative. So here's a picture down at the bottom. And what we've done is we've taken the bacteria and we've stained them, right? We've added a little dye. And the ones that have very thick walls, they absorb the stain the most, okay? And then they turn purple. So we call those gram-positive bacteria. The ones that are pink, they did not absorb as much of the stain, and we call those gram-negative. Now, in the past, gram-positive and gram-negative used to be used to classify harmful versus harmless bacteria, but we have found many different species that are now jumping the lines, and so we don't really use it as much. Gram staining is still an important technique because it teaches us about the cell wall, which is going to teach us about how we can penetrate that cell wall, if it is a thick wall or a thin wall. Remember, prokaryotes do not contain any membrane-bound organelles, so you will not see endoplasmic reticulum or mitochondria or chloroplasts or anything like that. So what do you see in a prokaryotic organism? Well, they do not have a nuclear membrane, but they still have the contents of the nucleus, right? We know that the nucleus holds the DNA, and actually in bacteria, there is a region within the center of the cell. While it doesn't have a membrane around it, it's still an area that is dedicated for this single loop of DNA that bacteria contain, and that is called the nucleoid, Okay, so the nucleoid is the region where the DNA is, no membrane. Bacteria can also have additional pieces of DNA. These are small, easily copied pieces of DNA called plasmids, and they're completely circular. And we'll talk about them more in a little bit. Also, all cells, prokaryotes and eukaryotes, have to have ribosomes because that is the basic machinery that it will be necessary to produce the proteins. And bacteria are living things. They need to make proteins in order to grow, to reproduce, to, consume food and digest nutrients and things like that. We know the cytoplasm is that fluid within the interior of the cell that sort of gives the cell shape, but even more importantly, contains lots of important monomers. Remember those building blocks for carbs, lipids, proteins, nucleic acids, and enzymes as well. Enzymes are very important to the interior environment. Other features that bacteria can have, flagella. So you're going to see these long whip-like structures coming off of the back here. Each one of those is called a flagellum. So multiple are called flagella, and they're going to whip them around and use them for movement. And then these shorter ones that are coming off all around the side here are called pili, or apillis for short. Not for short, I'm sorry. Pillis is singular. And what that's used for is that's used for cell-to-cell contact. So even though bacteria are single-celled organisms, they do associate with one another. And so in order to talk to one another, they're going to physically connect to each other. So they're going to plug in, and the pili are going to be important in that. How do bacteria reproduce? The main way that we're going to talk about for right now is going to be through binary fission. We know that bacteria reproduce mostly asexually, and what that means is they do not use another organism in order to reproduce. So when one cell divides into two identical daughter cells, we've called that mitosis, right? But when it's one cell organism dividing to form two identical celled organisms, that is called binary fission. So it's the same process, it's just that the results don't make two cells of a larger organism. Instead, they make two separate living organisms. Under, ideal environments, they can reproduce very quickly. Most likely when you get to college, you're going to do a standard population growth lab where you are going to measure how fast E. coli can double. And you're going to do that by looking at how cloudy your solution where the bacteria is growing will get. And in ideal situations, E. coli can double in number every 20 minutes. So they most definitely far outnumber us. Different types of environments. So we talked a lot about eubacteria, and we talked about how they live with us, on everything that we touch. There's bacteria covering everything you see right now. But there are also a class of bacteria called archaebacteria that are going to live in very extreme environments. So for example, thermophils are bacteria that thrive in very high temperatures. So here's an example of a hot spring, and the temperatures in here are, well around boiling or whatever. And those bacteria actually are designed, it's a weird word to use, but those bacteria are optimally functioning at that temperature. Okay, if you were to bring them down in body temperature, they would probably struggle. So think of a bell curve, right? Okay, and we have an ideal temperature, right? 37 degrees is our body temperature, and then we can, be comfortable between 100 degrees Fahrenheit and I don't know, maybe like 40 degrees Fahrenheit. But as soon as you start getting above or below that, we start having trouble maintaining homeostasis. And so we do things like we put on jackets to stay warm, or we start sweating profusely, or different things like that. Okay? Bacteria, they don't have as many bells and whistles as we do. So maintaining homeostasis is not something they do very well. Therefore, they rely on their environment to stay stable. So these bacteria, They're built to survive in these extreme environments. Another way the environment can vary is by the concentration of salt. Okay, halophiles are bacteria that can live in extremely salty environments. Okay, we know they can live in freshwater environments too. They can live in any kind, but some species are designed to live in these extreme environments. So, how do we use bacteria in biotech? Well, we use it to produce food. So, lactobacilli, we've already talked about this year in making cheese, making yogurt as well, and then also used in the pickling process, right? For making things like pickles and kimchi, which is a very spicy pickled cabbage. We also use bacteria in biotech because they are hands down the best machinery for producing protein. Okay, we need cells that are going to make a protein that we've designed by creating a gene. And so, what we do is we find the gene that we want, or we engineer the gene that we want, and then we insert it into a single bacterial cell. Then we culture that bacteria. So we're going to grow billions and billions. I mean, I went through a factory, that's not a factory, I guess you could say, it's a company where they make a protein product from bacteria. And I'm not kidding, the vat that was holding bacteria, the size of this classroom and 2 floors high. Okay, that whole thing was filled with a broth containing bacteria. So they let them grow and grow and grow. And then what happens is they kill them and they extract the protein, they purify it, they bottle it up, and they sell it. Okay. Most often we use this for things like making insulin. Okay. Diabetes is a really big problem, and we can engineer bacteria to produce human insulin rather than taking it from other humans or taking it even from other animal sources. So when we make a protein that has come from another organism like bacteria that we've genetically engineered, that product is called a recombinant protein. As I said, insulin is an example. Another one is cow growth hormone, okay? Farmers that use genetic engineering, which we don't see as much in the US because there's a huge movement against using genetically engineered organisms, but in some areas they do. So a cow growth hormone called bovine somatotropin, that gene was removed from cows. It was inserted into bacteria. The bacteria go through and make a whole bunch of that hormone over and over and over again. We extract the hormone, we purify it, we put it into a syringe, inject the cow with the growth hormone, and what happens? The cow is able to produce more milk and more meat. All right, so let's talk about eukaryotes. Eukaryotes, much more complex. They can be unicellular like bacteria, or they can be multicellular. So that's very important, a very distinct difference. They also include more kingdoms. They include protists as well as plants, animals, and fungi. Okay, and so just understand eukaryotes show a wide variety of life and a wide variety of strategies for successfully surviving. Their size, notice, is bigger. Remember, in bacteria, we were looking at 1 to 10 micrometers. Now we're at 10 to 100. So cells are much, much larger. And look at the variety of shape, okay? We see a neuron in the bottom corner. We see a plant cell up top. I'm sorry, the yeast cell didn't show up, but basically it looks like a big tan balloon that's been blown up. And these smooth muscle cells, each one of these little pink slivers is one cell, okay? So a variety of different shapes here. We don't have, 3 main classes or anything. They're far too varied. How do we use eukaryotic cells in biotech? Well, fermentation is really important. Yeast cells are going to be used in baking bread and in producing alcohols, okay? We've learned about fermentation in 9th grade. We know that as they are breaking down glucose, they don't have access to oxygen. And then as a result, they're going to go through a different pathway, the fermentation pathway, and that's going to create some different byproducts. And so in alcoholic fermentation, it will create ethanol, which is a form of an alcohol, and it will also release CO2 molecules. Both of these things are going to be used in the production of, alcohols like wines and beers and things like that, and then also in breads. Now, in bread, we bake bread, and so what happens is any of the alcohol that was produced through fermentation, that's going to be cooked off. But the CO2, that's what causes all those little bubbles and those little nooks and crannies that we love so much in our bread products. Also with biofuels, okay? Fungi are decomposers. And so what they're going to be able to do is they can break down cellulose. That is the name of a carbohydrate that is found in plant cell walls. We don't digest cellulose. Just think about it when you eat celery or if you eat corn, for example. Have you ever noticed that kind of comes out the same the other end? It's because our bodies can't digest that complex carb, but fungi are able to break that down. And so now, one of the things we're looking at are biofuels. Can we use fungi to break down these complex carbs in corn and then ferment them into ethanol? Because when you break cellulose down into its monomers, what do you get? You get sugars. And then if you feed that to fermenters, what's going to happen is sugar fermenting is going to produce ethanol, and ethanol is something that we use in our cars, right? That is our form of gasoline. We also use eukaryotic cells for recombinant protein production as well. So some examples in fungus, okay? We've extracted enzymes from the fungus Aspergillus there, and that's what is in laundry detergent. So when you get a protein stain on your shirt, I don't know, let's say a meat stain of some sort or something like that, the enzymes in your laundry detergent help to break up the stain. So it's not the soap is getting it clean, but sometimes, stains are results of proteins that don't break down. We use this enzyme from a fungus and we add it to the laundry detergent, and that's going to be sort of like a stain booster. Also, Chinese hamster ovary cells. Yep, I'm not kidding. So they are cells that are taken from the ovary of a Chinese hamster, have been used to produce antibodies, which are really important in making different types of drugs and medications. Also, stem cells is a very big area of research in biotech. We have some important laws here that prohibit use of certain stem cells. Mostly, we talk a lot about embryonic stem cells because those are taken from embryos, and those are the most controversial because an embryo will be destroyed by that. One thing I want you to be aware of is take a look at a pen and hold it up. And when they talk about the embryo that they are destroying, that they've cultured in a lab in order to do this, realize that they are talking about a tiny ball of cells, not differentiated in any way. There's no heart, there's no blood, there's no face, there's nothing like that. There's no brain cells even. There are just undifferentiated cells, and that could easily sit on the tip of your pen. Okay, so it's this little tiny ball of cells. That's what they're calling an embryo. I always think it's really important to point that out because when students hear embryo, they picture baby in the womb. No, absolutely not. Okay, when they're doing embryonic stem cell research, they are talking about little tiny balls of cells that could fit on the tip of your pen that are being grown in petri dishes. Okay, those stem cells are very important because, as we know, stem cells can differentiate into any cell type. Okay, so you can train them to become a certain cell. We also can induce pluripotent stem cells. So these are stem cells that are taken from adults. We do have a few different areas where we can take stem cells from our bodies. They've been differentiated a little bit. And so they can only develop into certain kinds of cells. But what we can do is we can try and induce them to sort of go backwards in their development so that they can differentiate into more types of cells. Still limited, but allows us to at least play with that. Why are stem cells so important? It's how we discover diseases, right? Because diseases are going to develop somewhere in the cell formation, right? Somewhere in cell development. And so as a result, what we can do is we can trace how a disease evolves, essentially. No, evolves isn't a good word. Let me think about a better word. We can think about how a disease is going to show up during development. Where during the splitting and the differentiating of cells do we see that mutated cell arise? Do we see that change occur? So it's very important for studying that. Also, if we want to replace damaged or diseased tissue with new tissue, we have to grow that new tissue first. Okay, things like that. So that's the end of this lecture. Key things you should be getting from it: differences in review from ninth grade of eukaryotic and prokaryotic cells, different characteristics of cells, eukaryotes and prokaryotes. Think about those classes, how they differ in shape and size, and then their uses in biotechnology. Okay, if you have any questions, please email me.